So good morning, everyone. My name is Shanita Hughes Halbert. I am a professor in the Department of Psychiatry and Behavioral Sciences and Associate Dean for Assessment, Evaluation, and Quality Improvement in the College of Medicine. And the AT&T Distinguished Endowed Chair for Cancer Equity at the Hollings Cancer Center, and co-program leader and associate director for education and training. One of the things that gives me great pleasure is to work with several investigators within our Precision Medicine Center, Minority Men's Health, and the Hollings Cancer Center, as well as colleagues within the Geographical Management of Cancer Health Disparities Region 1 South, to sponsor and coordinate a seminar series called DREAM, which stands for Disparities Research and Methods. And one of the things that we do as part of that, I have the great fortune of having nationally recognized experts in minority health and particularly cancer health disparities to join us and present their work related to cancer health disparities. So today we have Clayton Yates, who is a professor in the Department of Biology and Center for Cancer Research at Tuskegee University, to give a seminar on his work in immuno-oncology and breast and prostate cancer. Dr. Yates is an internationally recognized expert in prostate cancer health disparities research, cell biology and molecular pathology. He earned his PhD in the Department of Pathology at the University of Pittsburgh in 2005, and also received and earned a certificate of training in tissue engineering and regenerative medicine at the McGowan Institute of Regenerative Medicine. He's done postdoctoral training at Emory University in the Department of Urology. And from there, he went to Tuskegee University in the Department of Biology and the Center for Cancer Research. He has a very distinguished career and has earned several prestigious awards, and has really been leading our thought and research related to understanding cell lines based models that are derived from African American patients. So we're really fortunate and glad to have him spend some time with us today. So he has a number of extramurally funded centers. One is a research centers at minority institutions. He's site PI of the CTSA, which is being implemented collaboratively with the University of Alabama. And he's co-PI of a U54 Cancer Health Disparities with Morehouse University and the University of Alabama. So he's going to be talking to us today about his work in immuno-oncology targets. This is our first virtual seminar, so we appreciate Clayton being our first virtual presenter for our seminar series. So this might be something, I know it's going to go great, but if we experience technical difficulties, we'll work through it. So just to remind everyone, we ask you to put your phone or your line on mute, and then there'll be time for questions. And I think what we'll do, we haven't talked about this, but now that it occurs to me, is we'll use our chat feature, and so Clayton can respond to questions as they come in during the chat. Does that sound good?
Works for me.
Okay, great. Thank you. I'll turn it over to you now.
Well, thanks, Janita, for that very nice introduction, as well as inviting me. I know we originally planned to have this in person, and I would love to come visit your campus, but I'm also very happy to be able to do this virtually. I know many of us have had so many Zoom meetings over the last several weeks. And also, I would like to thank Erica for really organizing this and making it a seamless transition from being in person to online. So today I want to talk about how, basically, and give you guys an overview of our journey and disparities, how we started off as basic cell biology, and we kind of back end ourselves, I think, into immunology-based differences, and how that's now, transformed our thinking into how at multiple levels, how we address disparities, not only from really defining who are the people from a genomic perspective, but also really identifying things or actionable targets that we can act on sort of immediately. So, I can't advance the slide. First problem. Okay, there we go. So I do have a disclosure to make, and those are two here. I'm a consultant advisor for Riptide Biosciences, and I will be discussing development of an investigational drug that we have going into phase 1 toward the end of the talk. So, as we start to look at prostate cancer, I'm sure many of you are familiar with this. Prostate and breast are still the leading killers among African-American or disparate populations, particularly Black. And so, Franklin Hain published this paper, I think last December, or 2 Decembers ago, 2018 in JAMA, where I found this really, and I like to put this in every talk because I found it really alarming. And this is a very busy slide, but I want to concentrate on the red box here. So what the red box is showing that in a very large patient data head in the SEER cohort is that African-American men who present with low-grade Gleason 6 grade disease still die more frequently than other races. So, Gleason 6 is a low-grade indolent disease. Most men are placed on active surveillance. And so, really this suggests that there is not just associated economic disadvantage, but there's also a biological driver that's driving the tumors in men of African ancestry. So when we start to look at disparities, again, I like to put this slide is because we don't do it in a vacuum. There are multiple factors that all go into worse outcomes that we've seen, and so the aggregate larger data is that we have epidemiologically, anthropological, where we start talking about ethnicity, social constructs. Again, we'll touch upon some of that later on. But then, how is that all influenced through environmental lifestyles? And then if a person has a certain set of genes that are ancestry derived and they're in a certain environment that has these environmental lifestyle exposures, how does that, and then they may or may not have equal access to care, how do we sort of break down that? And so I think that we're figuring it out. There are a lot of people, including Chinita, who are leading in this area, but we still have a long way to go to start integrating the entire picture. And so, when we start talking about though precision medicine in cancer, this initial NCI trial, comes to mind, where the NCI initiated the precision medicine in cancer treatment trial, where they were genome sequencing over 10,000 individuals to see if they can match a drug to a proper mutation in individual patients. And so this is called the NCI MATCH trial. Again, it was 5,000 patients were screened and they were looking for matches. But one of the major, and there were some successes, but one of the major outcomes from this that I took from it is that not all patients with tumors actually have a drug. So this can be taken and interpreted several ways. It can be interpreted that there are novel mutations within the general population, or they're not capturing mutations specific to a population. And so there's a lot of work to be done, and this is the area that we have been focused on in our lab for the last several years. So when we start talking about genes are regulated by signaling, right? And so again, this is a really busy slide, but I like to put it up here because it's a lot going on. And so, our lab is, primarily when we started multiple years ago, we were focused on growth factor signaling. And we were focused on particularly the EGF receptor, tyrosine kinase signaling, and how that influenced cells to actually, lose their cohesiveness, break free from the tumor mass, and metastasize. And so if we started to look at the data, particularly this particular growth factor, EGF receptor, was, shown to be, this is a nice JCO article where they were shown to be 6-fold increase in African Americans. And so, again, if these growth factors, which are clinical targets, have drugs that have drugs available in the market for, if these are overexpressed, they're possible drivers of the aggressiveness that we see in African American tumors. And so other people have followed that up in a nice review suggesting that these EGF receptor, particularly these growth factors, are higher expressed in African American men, and then that correlates with the downstream signaling, the RAS-RAF-MAP kinase signaling, that it then influences increased proliferation, increased survival, increased migration, increased everything that makes tumors more aggressive. And our lab has done a number of work showing that if you use these inhibitors, that you can reverse this process. So we have been, really focused on this in a larger scale, and really started to narrow down, what are the signaling events that are responsible for this downstream mediated signaling from the EGF receptor, the growth factor receptor. So when we start to look at even more global, changes that occur and that have to go really fast if a tumor is progressing, they don't normally occur through genetic, they occur more through epigenetic. So we pulled this data from a group at USC, which did methylation analysis. And they used a gene called EK-herin, and EK-herin again is a cell-cell adhesion molecule that allows cells to, when it's present, cells are sticky, they don't metastasize or migrate, and when it's absent, they move. It's a clinical marker for many tumor types. And so when they looked at BPH samples, here is on the image on the lower left, is broadly expressed, and you see the cells are cohesive. But if you look in late stage primary tumors, there's a high percent of methylation, and the EK-herin is lost. But if you go back and look at bone met, so after the tumors that actually metastasized, they reform this cohesiveness. So and so I don't have time to show it, but when early on in our lab, we were one of the first labs, in addition to this, to show that these cells are actually going through dynamic changes in their morphology throughout the cancer progression. So it's not one-way street. The cells are changing based on the environment and sort of adapting to more from an epithelial to a mesenchymal phenotype. And so this really captured us that, if you were targeting an epithelial cell or a mesenchymal cell and it changed, the drug or the target wouldn't work. So we really started to focus on how these cells transition as possible targets. So we asked a basic question: what is the molecular machinery responsible for methylation silencing? And so just to back up a little bit on what DNA methylation is, it's the addition of a cytosine or a methyl group on the cytosine on the DNA. The reason DNA methylation is very popular is, one, is reversible. It's evolutionary derived, happens very frequently in during cell differentiation and embryogenesis. In fact, many of the events that we know during embryogenesis are methylation related, and it's been linked to lifestyle and race. And so a lot of groups now have started to show that African Americans have higher or increased methylation events. And so if you look at the central dogma of methylation, is that if a gene is unmethylated, the chromatin is accessible for transcriptional regulation, and then therefore genes are transcribed. If the gene is a methyl group sits on the actual DNA, it actually coils, and so then genes are turned off or silenced. And this has been the central dogma. However, there's actually a step missing in that dogma, is that there actually has to be an attraction of a methyl binding protein to that methyl group in order for the silencing to occur. And so this is a group here of multiple methyl binding proteins that actually are attracted to these methyl groups and then mediate the silence. So in essence, methylation alone is not important. It's not as a driver of gene silencing. And so our lab has been focused on one particular methyl binding protein called Kaizo. And so just as a sort of overview, because I know this is a bit complex, but if a gene is methylated, right, we typically associate it with silencing, but more and more result data has shown, including from our own group, is that Methylation is just a first step. A methyl binding protein actually becomes attracted to that methyl group, and that methyl binding protein complexes with it. Histone, deacetylases, NCOR, other cofactors that then form a complex on the DNA that then mediate the silencing of gene expression. So we also feel that this is a point of therapeutic intervention. And so just a little bit about Kaizo. The reason why we chose it, one, is a bimodal transcription factor. And bimodal meaning is that it just doesn't silence DNA through methylation. It actually has DNA consensus binding sequences, very much like all other transcription factors. So it can do it both ways, by sitting on the methyl group or regulating genes at the transcriptional level by directly binding the DNA. And when we started this multiple years ago, people had shown it, mostly in embryogenesis, that Kaizo was sometimes in the cytoplasm, sometimes in the nucleus, and it exhibited this cytoplasmic nuclear shuttling behavior, which we then interpreted that this could be responsible for how cells change their shape and phenotype throughout the cancer progression process. So our lab, and Jackie Jones, who is now an associate professor and assistant chair at Troy University, she was first graduate student, one of the first graduate students in the lab, came in and asked the questions, how is Kaizo expressed in prostate cancer patients? And so if you look here, it starts to become largely expressed in the cytoplasm in low-grade tumors, and shifts from cytoplasmic to nuclear expression and high-grade, and that continues to Mets. If you look at the bottom here, the Kaizo is particularly in the nuclear, where it could be binding DNA, is gradually increased as tumors become more aggressive. And at the time, we were just entering, I was actually just starting my lab. And so we knew there were disparities, but there wasn't really this field of genetic and gene changes. And so we actually asked the question, though, was there differences between African-American and Caucasian? And they were strikingly higher in African-American compared to Caucasians. And then because of the EGF receptor data that was very prevalent about EGF as a driver, we also performed in vitro experiments that showed that if you take cells that have largely cytoplasmic Kaizo, so this is here on the left, these DU145 cells, and the green is Kaizo, and blue is the nucleus. If you treat with EGF over a longer period, you can actually push it from the cytoplasm to the nucleus, all right? And this is just the Western blot at the bottom showing that we can reverse that. So we were then saying, okay, this could be fit into what we know it could be a driver of disparities. So Jackie went on to show that this not only occurs in prostate patients, but this also occurs in breast cancer patients. So this time we looked at over 600 patients, and we see that same cytoplasm and nuclear translocalization as tumors become more aggressive. If you look at primary tumor compared to Mets, if you look at the nuclear level of Kaizo in the primary tumor, if you look at the paired Mets, it's very, very clear that, in order to become metastatic, Kaizo is significantly increasing metastases. And then if you look at primary tumors versus Mets in Caucasian versus African-American women, you can see African-American women, primary tumors are equal to that of Caucasian women, and African-American women with breast cancer have the highest level of Kaizo. And then this was also followed up by Julia Daniels, who was actually the first who cloned the gene. She cloned it, and when she was a postdoc at Vanderbilt University, where she showed that not only in African-American women, she went on to validate this in Nigerian and women from Barbados, that Kaizo is much higher as well, and really highlighting its ancestral regulation in women of African ancestry. So again, because tumor cells progress and Kaizo could play a role in metastasis, Hong-Kee Wong from our lab, who is now an associate professor, who was a postdoc at the time, went on to show, to ask the question, is Kaizo actually functional and not just a biomarker? So we made Kaizo knockout cells, and you can see that if you knock out Kaizo, you can re-express EK-herin, and that's similar to even if you treat with a demethylation agent, 5-AZA. Kaizo re-expression, Kaizo knockdown causes re-expression of EK-herin and reversal of all mesenchymal markers. And more importantly, it changes morphology. So here is a picture where you can see, EK-herin largely not expressed in cells of mesenchymal. They have that fibrotic shape. But then if you knock out Kaizo, they become more epithelial and they form cells, suggesting they limit their ability to metastasize. And then if you look in primary tumors, you see a very similar thing as well. So again, ecadherin is expressed in green, and Kaizo is red, and you can see in an ecadherin positive tumor, there's very low limited Kaizo, and is expressed in the cytoplasm, but in a ecadherin negative tumor, Kaizo is highly expressed, showing that the cell line data replicates what's happening in human patients. So again, if you take this, and again, this was done by Balu Karanam, and who is Balu is assistant professor in the lab now, and Shana, who is actually at the FDA, went on to show that if you knock down Kaizo and you inject these into mice, the Kaizo knockout cells form smaller tumors, and they show a reversal of VMT, as you would expect that we saw very similar in cell lines. What I think is most striking, though, is that if you look at, if you can see here, is that, this is readily expressing Kaizo cells. So after 6 weeks, you can see they metastasize, particularly in the bone area. But in the ASH Kaizo cells, and we've done now this in breast and prostate, and so many different models, and even Juliet Daniels, as well as another collaborator, Kevin Gardner, has also validated that we do not see metastases in cells that have depleted or lower level of Kaizo expression, which we think is very remarkable. So just in summary of Kaizo, we're really excited about his work, and I'm sorry I'm having to move my pictures around so I can keep up with the slides. But Julia's lab, Julia Daniel, again, who we collaborate very closely with, she's at McMaster, and her graduate trainees really have focused on one of the other growth factors, TGF-beta, and how TGF-beta regulates cytoplasmic to nuclear shelling of Kaizo. And then our lab is really focused on how EGF receptor through RAS-RAF/MAP kinase signaling affects nuclear shelling to Kaizo. And essentially, this is a slide where we came together to share our data to show that both of these EMT promoting factors are regulating Kaizo, and Kaizo seems to be a central regulator of multiple signaling cascades and is a clear therapeutic target for limiting metastatic disease in both prostate and breast cancer patients. So just a little bit of transition. So during this time, a lot of work has started, particularly in disparities, has started to people have started really going very deeply. And one of them is centered around TMPRSS/ERG or TMPRSS/ERG expression. And so, the group at Michigan, Arul Chennai's group, has shown very nicely that TMPRSS/ERG was a common rearrangement in prostate cancer patients, and that developed, and then as a result, they developed this My Prostate Cancer Score, which takes into account TMPRSS/ERG rearrangement, serum PSA, and PCA analysis. And so there was a lot of work done, a lot of large papers. People have followed up on this very, very significantly, and they even came up with this nice cost-effective assay to measure prostate cancer progression. But as it turns out, as we now are knowing in many advances, is that a lot of these discoveries do not capture the specific genetics of all patients from different populations. And in this case, I like the title of this TMPRSS/ERG is not for everyone, because as it turns out, while TMPRSS/ERG is translocalizations are commonly identified in 50% of European patients, we only see about 10 to 20% of TMPRSS/ERG fusions in African Americans, and even lower in Japanese, Chinese populations as well. And then ERG, which is the byproduct of the TMPRSS/ERG fusion, is only expressed in 10% of African Americans. And so again, this is alarming, because while we have made advances, these advances won't help the populations that we're commonly or consistently trying to or used to have the most disparate burden. And so in order to start identifying that, Isaac Powell, I think he did one of the first and largest genome or genome-wide studies of gene differences in African Americans versus Caucasians. And so in this case, he did, I think about 650 patients. And at the time, he used microarray analysis, and it was a 590 gene microanalysis. And what they was able to show is ERG was lowerly expressed, as you can imagine here. This is red, as you can imagine, it has been widely reported. But when these patients who have, particularly African-American who have low ERG, they also have this increased inflammatory gene signature. So here you have IL-6 increase, IL-1B, IL-8, and there's some other ones, but I want to just highlight these genes as they're known inflammatory-based markers. And so I actually sent us this data, and so what we did was we went back in and we actually did a gradient analysis of these inflammatory gene signatures based on the quantile expression of ERG expression. So, you know, essentially on the left here is a low ERG to medium ERG in the middle and to high ERG expression. And then we overlaid IL-6, which represents an inflammatory response, and AR, androgen receptor expression. And so as you can see, as ERG expression changes from low to high, the inflammatory gene expression changes, and that concomitantly also changes with androgen receptor expression, right? And so at the end of the day, if you patients have low ERG expression, they have high IL-6 expression, but they have low androgen receptor, right? So again, this led us through all the data to come up with this sort of progressive diagram where African-American men who have low ERG expression, they have, you know, very few of this fusion status, they also leads to a low ARs. This low AR creates this stem-like feature in African-Americans that is associated with increased IL-6, chronic inflammation, and some of this chronic inflammation could be a driver of tumor genesis. And so, again, this was validated by Tim Rabick's and Schaefer's group in a nice JCO article where they showed that African-American men who have lost ERG, but also 3 other genes, SPINK1 and AMACR, also have this low androgen receptor expression as a driver of their phenotype. So, you know, now that we had a real, you know, this is a real problem, it's verified and validated in multiple patient data sets, we really needed to go in and figure out, you know, how to dissect these events. So our lab has been very, very interested for a number of years in establishing models of African-American prostate cancer that cell lines in particular that we can use to dissect our molecular events. So Shanice Theodore, who is at the CDC now, established these African-American cell lines in our lab. Many labs now have gone on to use these, and even Rick Kittle just published a paper, well, last year, February, showing that you know, these cell lines are indeed representative of African-American tumors because they have over 70% West African ancestry. I will, you know, suggest and just, you know, ask people, as you know, many labs are developing cell lines. This is a story where we had one particular cell line, E600, E006A, which was thought to be, you know, from an African-American man, and as it turns out, when Rick looked at the ancestry, it turned out to be actually very, very European origin and not African, and very little African ancestry origin. So it may be a good idea to characterize models before using them, or at least you know, understand their ancestral background, and so how we relate that to the tumor biology that we're seeing. So when we start to look at the biological process of our cell lines, you know, we saw very much that, you know, what was being, you know, very much like I saw in his patient data sets, is that cytokine, immune response, interference signatures were some of the most highly regulated gene sets. We also did a RNA sequencing of our own data set where we actually macrodissected the samples, and we saw the same thing: defense response to viruses, all immune-related responses were the most differentially expressed genes. So again, we saw our cell lines representing what we're seeing in patients, so we thought they were very, very, very useful. And so, you know, we start to look back at the literature. You know, it's been very widely. You know, I did the largest data set, but Renee Reams did the first data set back in, I think it's in 2001, where she did a microarray showing that the immune differences were the most significantly differential changes between African American Caucasians. And then Stefan Alm's lab followed that up again with again, a cohort of a separate cohort of patients from Maryland, showing very nicely that these immune genes are some of the top driver genes that are differentially expressed in African American Caucasians. And so Stefan went on, who was at the NCI, went on to show that it's not just, it's a group of immune genes, and it's a viral mimicry signature. It's a 47 gene signature that's upregulated in African Americans. You can see here African American prostate versus European, and they're related to an interferon-related DNA damage, or IRDS signature. So this is interferon STAT1 gene, and STAT1 is a really big driver of this interferon signature. This STAT1 interferon signature seems to be pervasive in African American tumors. And so if you look at the impact of this across the TCGA cohort, you can again see that, African American men have this, really unique gene signature, where a majority of the genes are overly expressed in African Americans, and that relates to a decrease in overall survival as well. So again, this immune signature appears to be very, very clear and present in African American men. And so our group was really interested in identifying, mitigators or why was this. And so in collaboration with Laura Suchison and Maury Campbell, who are at Ohio State, we use our African American cell lines to understand one of the more common factors known is that, people of African ancestry have low vitamin D. So we use our African American cell lines, in this particular case, 43 RC43N and 43T, and then LNCAP, which is Caucasian, and HPR1, which is Caucasian cell lines, and we treated these with vitamin D expression, right? And then we saw clear responses, and so we knew that they were responsive to vitamin D. And then we did RNA-seq. And what you could see here is that that the African American cell lines are much more responsive to the vitamin D compared to the Caucasian cell lines, right? And so this was actually vitamin D ChIP-seq, and this is actually vitamin D regulated genes, okay? And in both cases, if you do a ChIP-seq and overlay it with the differential gene expression, or you just look at, responsive genes to vitamin D treatment, in both, in more particularly in the non-malignant cells, you can see how African Americans are much more responsive. And then if you take these gene sets again and you do a GSTA enrichment analysis back to the inflammatory immune signature, so we see these changes occur as a result of a change in inflammatory immune signature in both, even the non-malignant and malignant cells. And if you then start to narrow down some of the most differentially expressed genes that are responsible for this inflammatory gene signature, particularly AR, BDR, we came up with several two genes, and I want to highlight two, BAZ1 and SMARCA5. And so in this case, we went back again to the TCGA data set, where we saw that if you look at BAZ1 and SMARCA5, you see significantly differentially expressed in African Americans versus Caucasians. So we followed up these two really interesting genes. So if you can, treat with, look at the African American cell lines treated with vitamin D expression, again, you can see BAZ1 and in this case, we haven't gotten to SMARCA5 yet, but the BAZ1 is very responsive. And if you look at the correlation of this, a BAZ1 and SMARCA5 expression into an inflammatory, and surprisingly, and this is something that is very new for us, and we're really excited to follow up. In fact, we just, Maury Campbell now, we just got a DOD grant funded in the last study section Health Disparities Award to follow this up, is that these genes seem to be mediating an inflammatory based circadian rhythm change or epigenetic change in African Americans in response to vitamin D that is not occurring in Caucasians. You can see this too. If you look at the ChIP-seq data here on the right, and this is again a volcano plot, see how the African Americans have more gene expression changes than the Caucasian cell lines. So there's actually a greater response in African American cell lines treated with vitamin D compared to Caucasians treated with vitamin D. All right, and so again, we're suggesting that this is due to the inherent lack of vitamin D, that African Americans are actually more responsive or receptive to vitamin D treatment. And so, as you well know, and I have to admit that a lot of this work was guided by a seminal publication from Chanita's group, where she did she genome sequence or RNA sequence. I think it was 25 or around 20 or so African American Caucasian men with prostate cancer, and then prior to prostatectomies, they treated with vitamin D for several months, and then resequenced these patients. And when they saw a complete reversal of the immune signature, so again, in collaboration with Chanita, we actually went back to look and see if any of our genes that we were seeing in our cell lines actually were mimicking or being regulated in the vitamin D treated patients. And so here's some new data that we was just generated. We're trying to finalize a manuscript on it now. But again, as we saw in the cell lines, African Americans are here in red. And so the African Americans actually, again, are more responsive, very much as we saw in the cell line data. And there are Europeans who weren't really responsive to the vitamin D at a transcriptional level, right? And if we take those gene expressions, and I'm not only showing you the African American response here, we can really see how the African Americans, again, this is a basic gene enrichment analysis, and at the top here are just 3 different gene enrichment data sets and all the genes that contribute to the gene signatures. We really see that the vitamin D treated African Americans go from that immunosuppressive to the immunoactive, right? So we see TNF alpha, NF kappa B signaling, which we know is anti-tumor instead of that pro-tumor interferon gamma signature that we were seeing from Stefan and other groups in our cell lines. So we're really excited that we're starting to dissect not only that vitamin D is useful, but also how and why it's useful, and particularly in African American men. And so, in addition to our work that I do in the lab, I actually co-direct a prostate cancer transatlantic consortium with Falaki Odedna. In fact, this is where I met David Turner, and we had a very good time in one of our first meetings. And so I only say that is to show us that Stefan and I are now doing what I think is going to be a very impactful study, along with Maury Campbell's group, where we're genome sequencing African ancestry men, Native African ancestry men, and correlating with inflammatory gene signatures in the blood, as well as vitamin D levels, and doing multi-level omics, RNA exome, and DNA methylation sequencing on over 350 patients, as well as comparing serum analysis in over 3,000 men of West African ancestry. And so we're only in year 2 of that, and I do have some small data that we're writing up for publication, but we're really excited, and hopefully I'll come back and share the results of this. But we really feel that this is going to be a useful tool to start dissecting the roles of an influence of this mechanism in a larger cohort data set. So for the last part of my talk, I'm going to switch to our work in breast cancer. And so, as again, breast cancer, as many of you know, used to be typically associated with, Caucasian or European women having an increased incidence. However, as of 2012, actually African American women incidence has actually caught up, so it's almost equal now. And again, the mortality rate is significantly increased in African American women compared to European women. And so, if we talk about breast cancer is a cancer of different subtypes. And so, the basal particularly light subtype it has the worst outcome compared to a more luminal subtype, which has a more favorable outcome. And so, our lab, being a primarily prostate cancer lab, asked the question, what if we add an androgen receptor, or look at androgen receptor in the context of breast cancer patients? So as many of you know, androgen receptor, again, has typically been associated with prostate-driven hormone. But as it turns out, AR is actually commonly expressed in low-grade tumors, but only about 10 to 15% of triple negative, which is, again, the more aggressive phenotype. And if we look at inherent data here, AR negative patients actually do worse. They have a worse survival compared to AR positive patients. And if you look at predominantly a large amount of the cohort, there's a number of patients that have shown, manuscripts that have shown that a majority of actual what we're calling triple negative or AR, ER, ER/PR and HER2 negative are also simultaneously androgen receptor negative as well. So we asked a simple question. Wendy Dean Colon, who is an oncologist, asked a simple question in our group. What's the AR expression? What's the distribution of AR in African American versus Caucasian women breast cancer? So this is the TCGA analysis when we looked at AR in white women is decreased only 56%, while 81% of African American women were negative for AR in our cohort. In the TCGA cohort. And then not only were they negative, if you looked at an AR negative patient, particularly African American compared African American to Caucasian, AR negative African American women particularly diagnosed earlier in disease, around age 49, compared to an AR positive patient, right? And then in African American women, and then this similar trend was also observed in even European women, where age 50, if a woman was age 53, they were diagnosed at, AR negative, they were diagnosed at age 53, compared to an AR positive patient diagnosed at around age 55. So again, as we know, triple negative African American women are diagnosed at a younger age. This fits the sort of overall data that we knew about African American women being diagnosed at a younger age. So Melissa Davis at Cornell went on to show that if you take these AR positive patients and the AR negative and dichotomize them, and you overlay that with the PAM50 analysis, and again, the PAM50 gene set is just the way to determine the breast cancer subtypes, luminal versus basal versus HER2 versus normal. What we can see is that these AR negative patients have, again, an enriched basal-like phenotype. And remember, as we showed before in the earlier slides, that the basal subtypes have the worst outcome, right? And it seems to be associated with these AR negative patients. So again, we went on to determine that these AR negative patients, again, are predominantly basal. So if you look at, even white or your African ancestry, if you're AR negative, you're basal, you have over 70% chance of having a basal subtype. And if you look at these African American women with the basal subtype, and this is a cumulative progression analysis. So what it's suggesting is that African American women with AR negative basal-like tumors progress faster through the progression of disease than if they were either Caucasian or AR negative. All right, so these AR basal-like subtypes are really the fast progressing tumors and ones that need to be monitored the most. So again, Anusha and Essence in the lab went on to show in a separate data set from a partnership that we have with Caris molecular diagnostic testing, is that we were able to take over 800 patients and validate that over 79% of what we were calling triple negative were actually AR negative, right? And so, and then over 75% of the metastatic or recurrent samples were also AR negative. And so Essence did what I think was a really interesting analysis. So what we wanted to determine that if a patient was AR negative and the tumor actually, and a second biopsy, or the tumor actually metastasized or recurred, were they still AR negative, or would, or did they change, or their subtype changed based on, a second biopsy, or did they actually metastasize or recur based on treatment? So again, if a patient was initially diagnosed with a luminal A, 70% of those patients retained their luminal A subtype. The same with HER2 enriched. But if you look at triple negative, it was only 50% of triple negative women actually retained their triple negative status in a subsequent biopsy, okay? But if the patient was initially quadruple negative, 74% of those patients retained their quadruple negative status. So again, we feel like more quadruple negative, or an addition of AR as a diagnostic in addition to ER, PR, HER2 status, is useful to determine those really aggressive, stable phenotype tumors. And so again, if we look back in the TCGA, and again look at African American white versus Caucasian, we can again see that these African American women have these overwhelmingly basal, this case basal-like 1, basal-like 2 immunomodulatory subtype compared to a Caucasian women who have a more mesenchymal, sort of mesenchymal subtype, all right? And so again, now starting to dissect these features is really important in us understanding how to treat this patient population. I want to add, and this is a manuscript. Actually, it was just accepted this morning, so I put a slide in. Is that we've gone back in and resequenced some of that work before was in the TCGA. We've gone back in and resequenced our own data set. And then this time, and instead of just asking if there was self-reported race, we actually did qualified genetic ancestry using the RNA-seq data on each patient, and then analyze their differential gene expression. So here on the left is their qualified gene ancestry. And so we use over 70% West African ancestry as a cutoff. And then we perform heat maps. And again, it's actually out in preprints now. So I put this link here at the bottom in case you want to see it today, but it was accepted formally this morning. And so again, if you look at European Americans versus African American women, you start to see really clear differences. That, a lot of times when we do self-reported race, and it's actually in the supplemental data of this manuscript, we see a sort of mixture. We see differential genes. Based on ancestry, we start to see a much clearer picture. And in fact, what we see in European patients here in this blue area is that many of these patients are very admixed, right? So they have an admixture of African ancestry and European ancestry compared to just African ancestry or just European ancestry. And then when we do a phylogenetic tree, you can start to see the separation, right? So these red dots here are suggesting that European Americans all are connected through a phylogenetic background, where if you look at the blue dots and the branches from the blue dots showing that the African American patients, again, based on African ancestry, are all connected in phylogenetically, right? And so again, we were really excited about this. And so now we're starting to use genetic ancestry, or at least qualified genetic ancestry, to separate our data sets. And so Shakir Muhammad in our lab went on to show that if you not only look at the gene expression through qualified ancestry, but you also look at the exosomes. So exosomes are these secreted microvesicles that come from cancer cells. If you look at these exosomes, or the gene expression in exosomes, we also see this inflammatory immune signature, right? So again, this is African-American on 14 versus European. They were all matched controls. So each one of these were matched to their normalized controls. And what we see again is a clear change in the overall gene expression profile, and robustly in the innate as well as adaptive response. And so while this is a busy slide, I want to sort of call your attention to the sort of dots here. MAP kinase signaling, NF-kappa B signaling, JAK-STAT cytokine receptor signaling. The major genes that we showed in prostate, we showed in breast gene expression, are also expressed in secreted vesicles that are communication vehicles for how cells actually all get on the same page in the microenvironment. And so we're starting to see this compound of data at a multi-level, and multiple levels. And so, again, we're a primarily based molecular lab. So what Shakir did, he went on to take that patient data set and overlap it to kaizo-regulated genes. And again, what we found was very, very interestingly, was that there are a number, in particular our case here, a gene set of what we call inflammatory-based genes that were overexpressed in African Americans, but were also linked directly to kaizo-driven gene expression. And so the two that we pulled out was CD47 and TGF-beta1, which we know were very, very important as drivers of kaizo expression. And so again, in this case, as we did in the prostate model, if you deplete kaizo, you have very low tumors. And in this case, we looked at CD47, which is the don't eat me signal, is almost completely lost in the kaizo depleted cells. SERPA alpha, which is the ligand for CD47, is completely lost. And thrombospondin 1, which is a tumor suppressor gene, is also re-expressed in the kaizo knockouts, tumors from the kaizo knockout cells. And in addition to these, we saw that there was also change in the macrophage population. So in the kaizo high expressing cells, M1, which is again is a good macrophage. These are cells that actually go after and tackle tumors. Are lowly depleted, but they're highly expressed now in the SH kaiser cells. And CD206, which is the M2 marker, which promote tumor suppression, are very decreased compared to the scrambled cells. And so we've been really excited about this. And so that we saw in vivo. So we did what I think was a very straightforward and simple experiment. We took exosomes, secreted from 231 cells, and we put up and we basically treated MCF7 cells, which is a non-metastatic cell line, and we wanted to see if we can actually influence the macrophage polarization. So could we make good or bad macrophages? And just by taking the conditioned media of that, we can see that there is an increase in, if you take, Kaizo exosomes from Kaizo knockout cells, you can get a more, tumor, anti-tumor response versus if you take, you get a decrease in the sort of tumor suppressive macrophages. And then if you actually take these macrophages and you apply them to cancer cells, and this is, again, a very busy, green is the THP1 macrophages and red is the 231 cancer cells, you can actually start to see macrophages actually start phagocytosing the cancer cells in the absence of Kaizo, right? So we were really excited about this data. But we wanted to follow this up in human patient sample sets. And so in collaboration with Kevin Gardner, we did multispectral imaging on breast cancer patients. So in this case, we did 4 or 5 marker analysis, CD8, which is T cell, macrophage, a pan-macrophage marker, which is CD86, and then PD-L1, which is we know is immune checkpoint inhibitor expressed on tumor cells. And so we did a co-staining, and we compared that to this place, cytoplasmic Kaizo. And not only do we see the differential chain expression, what Kevin did, which was really very, very interesting, he did nearest neighbor analysis. So what we wanted to see is that if a cell was positive for Kaizo, how close was a macrophage? Was a macrophage much further away, or did it actually migrate toward the tumor cells? And what we can see is that African-American women expressed here in red have a much, in their immediate microenvironment, have an increased number of CD8, CD68 positive macrophages compared to Caucasian patients. And again, this manuscript is under review in Nature Communications. So just in summary of this part of the talk is that, in addition to the macrophages, we sort of derived what we think is a signature that's associated with a macrophage immune signature, where Kaizo is a driver. CD47 is highly expressed in African Americans, not as much in European Americans, and as well as CD47's ligand SERPA alpha. So we start to see this whole, sort of unique gene signature, and I don't have the time to show you all the data. This is also associated with a lower survival and outcome in these patients as well. So again, in just summary, so we have macrophages, which again is expressed in the microenvironment. So if exosomes are from a Kaizo high expressing cell, we see those macrophages polarized to the M2, sort of the bad macrophage, right? So SERPA alpha secreted, it upregulates CD47, and it escapes immune response. But if these cells are exosomes are secreted from Kaizo low cells, we see them more polarized to more of an M1 response, and these M1s actually participate actively in immunity to eliminate cancer cells. And so again, we're starting to see this very, very clearly as a disparity associated pathway. And this is really important because CD47 actually is one of the newest immune checkpoint inhibitors that have entered clinical trials in the last year or two. CD47 is again, an antibody came out of Herb Weizmann group at Stanford, where it's shown very nicely that, anti-CD47 is very, very important therapeutic. And, we're making a case that African-American should be included in, particularly in these clinical trials. And so for the last bit of my talk, I'm going to end very quickly, is that not only do we see CD47, as a result of these studies, we actually really dove very deeply into this macrophage biology and to develop a therapeutic, which we feel is very, very important and should be entering clinical trials later this year. So, again, just a bit about the macrophage biology. Here you have a macrophage that can be polarized to an M1 response versus an M2 response, which has, this THP2 response, which is immune suppressive, versus the immune promoting M1 response. And this is really a big driver of tumorigenesis. As we know now, many checkpoint inhibitors even fail because they have this Th2 response that cannot be reversed. And so our group was able to develop what we're calling a small molecule or small peptide inhibitor to one of the markers of macrophage in CD206, where we're able to use CD206 as not only a marker for M1, M2-like macrophages, but treating with CD206 rapidly causes repolarization of those M2 cells back into an M1 phenotype, and they participate in immunity. And so here's the data. So if you look at, and again, this work was actually just published in Science Translational Medicine in February, where if you treat with our inhibitory peptide, and we're calling RP182, you can actually repolarize macrophages from their M2 phenotype to an M1 phenotype in as little as 24 hours. Okay. And in addition to that, I don't have the time. It's a very huge manuscript with just tons and tons of data. But just as sort of a summary, if you treat with these these macrophages and targeting CD206 receptor, we're able to uncover a whole signaling cascade, ultimately ending with activation of NF-kappa B that causes secretion of TNF alpha, and the secretion of TNF alpha initiates the immune response and causes T cell infiltration, right? And so you can see that here. If you look at these, these macrophages that were treated with the peptide, you see robust increase in TNF alpha, IL-1B, lower than PD-1, lower than PD-L1. And as I mentioned before, SERPA alpha, which is the ligand, is almost completely gone. And if you look at the response, in this case, we use pancreatic cells. If you look at response to standard care chemotherapy, RP182 lowers, these are P16 transgenic, P16 KRAS transgenic mice. You can see they lower tumor volume in these mice compared to clinical standard of care, gemcitabine, which was at the time. And in combination, it just really, really, blocks tumor growth. And not only does it block tumor growth, we see a very nice increase in tumor survival. If you look at this in PDX, so PDXs that have high CD206 versus PDXs that have low, they're very responsive to our drug. If the patient or the PDX did not have any CD206 or very low, they weren't responsive. So we see this is very CD206 driven. And not only if you look at tumors from this, we get this M2 macrophages decrease, but you see a robust increase in CD8 positive T cells and decrease in Tregs, which we know again is a reversal of the tumor macrophage, the tumor suppressive response. And you can see this at the histochemistry levels too, right? So these RP182 treated tumors have very nice infiltrating T cell activity when compared to control cells who we really don't see any T cells, right? And if you look at the macrophage to T cell ratio as well, we start to see that these macrophages are rate limiting factors, whether T cells, particularly CD206 positive macrophages, are rate limiting factors to whether T cells, CD8 positive T cells infiltrate or not, all right? And then again, if you look at this in combination with PD-L1 therapy, we see a very similar effect. In fact, most of the world is, trying to understand how you can have responders and non-responders with PD-L1 treatment. We see that the combination of our CD206 inhibitor with PD-L1, it gets those non-responders, where you see here, to actually respond to respond to therapy. All right. And again, just to show that it's not just T cell response, we actually use a CD8 inhibitor to block the T cell infiltration. And again, we see that if you block the T cell infiltration, is not as robust as a response as you will T cell infiltration, but you still see blocking the macrophages alone is an important mediator of response, particularly targeting CD206. And so again, the proof is in the pudding, right? So what you can see here, this is a phagocytosis assay where you can start to see, again, green labeled macrophages start to engulf cancer cells, right? So we can see all these red circles here are engulfed cancer cells. And I know it's a little hard. These are KPC and PANCO2 cells treated, and I know it's a little hard to see, but I have a nice little video here where you can start to see that a macrophage, right, that was treated with the peptide actually can hold on, it's coming there. All right, so it starts to sort of gang up on it, and it starts to phagocytose the cancer cell, right? and then you see the green diminish. And again, we see this very nicely in the histology and then by EM as well, right? So, you know, RP182 targeting CD2 makes these very functional. So just in summary, you're right. So, you know, all of immunity is, you know, particularly checkpoint inhibitors, immune therapy has been about targeting or activating T cells. We feel like the next wave of cancer immunity is macrophage biology. And so macrophage biology is a rate limiting factor. In fact, we have identified CD206 as a new macrophage checkpoint. We named it in the Science manuscript. And this is, again, we see this very, very useful in combination therapies with checkpoint inhibitors, particularly in patients that don't respond to conventional therapy. And again, we're currently developing the Kaizo inhibitors as well. But as you can imagine, checkpoint inhibitors and immunobiology has really taken forth. And so we've really started to lead this toward clinical trials. I wanted to end by saying, you know, our lab is really focused on health outcomes. You've probably seen a number of activities over the years. We've changed and become bioinformaticists and now even drug development lab, because we're focused on health outcomes. You know, we know that many factors complete gene expression. There's so many changes. We see ourselves as not single focus labs, but as, you know, people who like to bridge and collaborate with a number of folks to get to what we hopefully would, at the end of the day, was positively affect disease and decrease bad health outcomes. And so, again, a number of collaborators have contributed to this work, and I think diversity starts in the lab, and so we like to keep a real diverse group. And so these are the people who do the work, and I thank them very, very nicely, and at this time, I'll take any questions. Thank you.
Thanks, Clayton, for a great presentation. We have a couple of minutes for questions, and what I would ask is that if you do have questions, please type them in the chat box, and probably maybe just to facilitate the process, you can type your questions and so send it to everyone. Are there any questions for Clayton? People might be having, I guess if folks might be kind of, may have a little bit of difficulty with typing a question in. Are there questions that people would want to ask? You might want to take yourself off mute and just ask the question.
Yeah, I think we have about 20 people. We can take off mute. Probably handle it.
Are any questions for Clayton? Oh, so there's one question. Is there any concern for antigen sync as they are observing with CD?
You broke up, Janita.
Clayton, do you see the question in the chat box about antigen sync as they are observing?
I can't get to the chat. Okay, stop sharing. Maybe I can get it here. Let's see what the chat box comes from.
So I'll read the question. It says, is there any concern?
Okay.
Yeah, do you see it?
So yeah, in the CD47 trial, so that was a problem initially. So they actually formed another group. There's so many groups now who have really delved into the CD47. They've actually eliminated the FC gamma region and from the original antibody constructs, and it seems like they've gotten around it. But that was a problem causing a little bit of toxicity in that original CD47 antibody from Irv's group. But other groups now have come in and gotten around it.
Okay.
Hey Clayton, how are you doing? It's Dave.
Hey David, how you going?
Not too bad. Good to see you, mate. Great talk. I was wondering if you'd looked at where Kaizo is expressed in the stroma in your primary tumors.
Yeah, so
That was my exact
we
question.
did see it expressed. Yeah, so it's low level expressed in the stroma, not as much. It's expressed on endothelial cells. We're starting to see it in lymphocytes a good bit, and so we're, you know, as we're now getting into the immune work, we see it expressed on lymphocytes, particularly in the tumor cells. Not as largely as much expressed in like fibroblasts, you know, if you will. So, but endothelial cells definitely, and immune cells, particularly lymphocytes.
Yeah, I ask because with the ages that when we treat them, we're seeing a big upregulation in the fibroblasts of KASO. And we're seeing that we get a more activated phenotype, cancer-associated fibroblasts, things, upregulation. We're also getting effects on the macrophage as well. So after this talk, we'll have to go back and have a look. But it's definitely upregulated in the fibroblasts in our model, in the breast cancer model.
Regulating. Wow. Okay. Interesting. Oh yeah. So that was the same question from Lillian Harris. Yeah. Right.
So I'd just like to bring us to a close by thanking Clayton for being our first virtual seminar presenter. We hope that it was a great talk. Hope that everyone was able to log in and see the Zoom and all the things okay. We'll go ahead and bring our seminar to a close. One thing we are going to start doing is just sort of a post-seminar evaluation, so we can make sure. Yeah, you can take your lunch break. It's lunchtime, sorry. Is what we'd like to ask you to do is to just complete a really quick survey to help us understand how we can improve upon, you know, what we offer in terms of topics. So we'll send out a short evaluation within the next few days and ask you to complete that and return it to us. The other thing I just wanted to announce, just in terms of our formatting. So we're really excited when we can have national, international experts join us and share their work. And we really appreciate them taking the time to do that. What we will do moving forward is sort of alternate our schedule with having a journal club format. So next time that we'll convene, it'll be a journal club that addresses an issue and research and methods related to cancer health disparities. So we're really excited about that and more information to come. So just in conclusion, thank you, Clayton, for a fantastic talk and for spending time with us today. And thanks everyone for taking time out of your schedule to join us for this virtual seminar. Thanks everyone.
Thank you. Bye-bye.
Thanks, bye.
Thank you.